See, we are recording, which is good. Someone just hit that. Thank you. Hope everyone had a good weekend, though. Before we get cracking today, any questions from last time or any of the other content that we've talked about before?
Feel free to either speak up or
Yeah,
drop a question in the chat as well. So I'm
yeah. I was going to
monitoring the
say it's
chat.
perfectly fine if not, and obviously feel free to drop anything in the chat or reach out to us via email afterwards as well. That's totally fine. But as you can see here, I'll get going. But we're going to be talking about gene editing today. More specifically, we're going to kick off with a couple of slides surrounding stem cell disease modeling workflows. This is always just a nice thing, I think, to do in a course like this to review a bit of the content that we've touched on in the past, and also serve as a nice segue into gene editing, kind of tell a nice comprehensive story, hopefully. But we're going to spend the bulk of our time today covering two things, right? The first is in regard to some really cool technology surrounding designer engineered nucleases, more specifically CRISPR and TALENs. And then, as I often like to do, I thought it'd be interesting at the end to put some of this content into a case study. And I'm going to highlight a gene editing cardiac disease modeling story. It's a 2D cardiac disease modeling story. I think it's quite cool. It highlights a bit of the work that we, Thermo Fisher, as a company, has been doing in this space. But I also picked it because I think it's a nice segue for next time and help set the stage a bit for Omar and Dr. April Pyle from UCLA, who are going to be talking to us next time around more specifically about differentiation, mesodermal differentiation at that, which of course includes the heart. So I always like to start with this slide. Again, it's a nice overview of the stem cell disease modeling workflow, and I'm a big visual learner, so for me, it helps always put things visually into context from a high level, kind of piece together all those buckets of the workflow. We always start with a particular cell model on the left here, right? We isolate somatic cells, for instance, from a donor. We then reprogram them through a variety of means. Omar talked about this before, right? To become induced pluripotent stem cells, or iPSCs. Nowadays, we often engineer those cells, right, to program target nucleases to, say, specific sites in the genome, which of course we're going to be talking about today. And then we've emphasized this before, right? But when you're challenging the cells with things like reprogramming or gene editing, which put quite a lot of stress on the cells themselves, it's especially important to routinely characterize the cells because obviously this gives you, as the researcher, good peace of mind as your experiments are progressing. But you have to take stock with the cells as well. So, right, you've got to check in with them. You've got to confirm their cell identity, their genomic stability, their pluripotency, and so on and so forth, right? As we've talked about before. Nowadays, again, these cells are often differentiated, though, into physiologically mature germline specific cell types, right? That, of course, allow us to, nowadays anyway, better mimic relevant disease states. And again, we're going to do that today in the context of a cardiac disease modeling story and showcase a couple of the downstream tools that we use to assess those different cultures. And so, along those lines, I always start with this slide as well. I think good cell models are really, really important, right? You've got to think about this from the get-go. What are you trying to model and how are you going to do it? And a good cell model, right, first and foremost, has the capacity to predict an output that translates to the organisms that you're studying, right? In other words, is it physiologically relevant? In our case, with regards to human biology. And there's a lot of different types of outputs, right? They come in a lot of different flavors, right? We've got gene expression, protein function, cell health, cell phenotypes. The list goes on and on. I've only listed a couple here, but basically it's from these various inputs across the top here that we're able to generate all these different data or outputs, which of course we then decipher and then we translate back to the organism. Again, in our case, as selfish as it often sounds, making sense of all of these different results that we get and translating them back to the human state. Now, Omar talked about this before in the second lecture as well, but with the advent of iPSC technology, there's been some really big advances in the workflows that we use that allow us to create even better models that, again, correlate well to human disease. Nowadays, even honing in on the contributions of, say, a single gene with respect to a particular disease. And again, we're going to do this at the end of today's talk in the context of a heart disorder called dilated cardiomyopathy. And so once we have all those cells for in vitro modeling, right, the next step in our workflow is reprogramming, which again, Omar covered in the second lecture. And again, as a quick refresher, that's that process of reverting a somatic or adult cell back to a pluripotent state, right, so that it behaves like an embryonic stem cell. And we do this by forcing the expression of four different transcription factors, right? OCT4, SOX2, KLF4, and c-MYC. And of course, as we talked about, there's loads of different ways to deliver these factors into cells to start that process of reprogramming. And along the way, we've got to give really important consideration to things like safety, consistency, workload, cost, the cell type that you're reprogramming, right? When you pick which technique that you're actually going to use. And as you see here, we're spoiled nowadays, right? There's loads of different choices or different techniques at our disposal. But again, from a bird's eye view, most reprogramming technologies deliver these different transcription factors through either integrative or non-integrative methods with those traditional technologies, right? Integrating into the genome of target cells versus, say, the non-integrating methods, right? As you see in the green boxes here, flanking along the edges, that allow us to generate iPSCs that do not contain things like detectable vectors or transgenes. And in our hands anyway, the most efficient of these are sendai viral particles and episomal DNA vectors, which I've circled here in red, and both of which we offer at Thermo Fisher to support reprogramming of a wide range of somatic cells. So again, continuing on, right, within our stem cell workflow, gene editing is really the meat and potatoes of what we're going to cover today. And so I always thought about this, and this is how I learned it in many moons ago. When we're editing text in, say, a Word document, right, we're adding, removing, or replacing words or paragraphs or sentences, right? And so you can think of genome editing in the most simplistic of terms of an approach in which the genomic sequence is directly changed by either adding, replacing, or removing DNA bases, right? But we also have to think about, in our case, in the human body anyway, the genome is quite resistant to change. DNA in our body is, of course, not only responsible for encoding all the necessary functions of the cell, right? But it's also quite crucial to determining differences between you, me, and all the other individuals out there. And so if DNA was quite easily altered, which is not, a lot of cell functions, right, could be disrupted, often in undesirable ways. And so what these organisms, and humans being one of them, have done to protect against these inadvertent changes is develop these inherent mechanisms that allow us to proofread and repair the genetic code, right? And so that's what we actually tap into when we're performing genome editing. And when we use genome editing to introduce different desired phenotypic traits into cells and organisms, right, what we then do is create much, much better models within a whole wide array of applications, as you see here. And I've listed a couple, right? Drug discovery, cancer research, epigenetics, functional genomics. Genome editing is used quite broadly nowadays, and it really has changed the game in as far as what we're actually capable of doing in this space, right? And, you know, kind of along those lines, with any scientific application, right, there's been some pretty hefty advances over the course of time, as you see here, right? With genome editing, for instance, from its early conception all the way through to the advent of different technologies like CRISPR-Cas9, there's quite a rich history involved and some really important and quite clever people involved in those steps along the way that have allowed us to evolve this process from really one that was limited to, say, a basic research setting all the way to a whole host of different applications in medicine, agriculture, right, scientific research and beyond. And so I pinched this. This is a general timeline from Nature Protocols from 2016. So it's a couple of years old now, but it's a cool paper, kind of highlights things out in a nice stepwise manner and talks on some of the technology that you see here on this slide. Things like yeast models, zinc-fingered nucleases, all the way through to more recent advances, right, like designer engineered nucleases, which again, we're going to be talking about shortly. And there's a lot of different angles that we can take nowadays to genetically engineer those stem cells, right? Things like gene repair, overexpression, the addition or deletion of certain genes. And again, we tap into that. We take advantage of that to help us build even better models, as you can see across the bottom here, and then again, investigate different disease mechanisms to understand human biology. This slide, again, just a nice general overview, talking on some of those current strategies underlying genome editing, whereby we're doing things like inserting defective genes to generate disease models, maybe inhibiting certain genes to, say, render disease-causing pathway inactive. We might use them in the context of drug development, right? We might insert, say, a drug-sensitive marker that allows us to do something like test the safety and efficacy of a drug. The list goes on and on here. There's loads and loads of applications nowadays, but gene editing is a really, really cool space, one that's rapidly evolving as well, and seeing its kind of fingers get into a lot of different applications. And so I think a lot of these capabilities nowadays are absolutely just incredible. But as good a starting point, I think, as any to talk about gene editing in a bit more detail, again, is designer engineered nucleases. And for us, this includes strategies like CRISPR or TALENs, which are designed to target nucleases to very specific sites in the genome, and then essentially create double-stranded breaks at a desired location, as you see here in the top with this red arrow. Following those double-stranded breaks, what happens, as I mentioned, is the natural repair mechanisms of the cell actually heal that break. And this is often done by techniques like homologous recombination or non-homologous end joining. And again, we're going to go into a bit more detail on what those mean in a moment. And so, as I alluded to, I want to talk today about two big technologies, CRISPR-Cas9 and TALENs, right, that again allow us to program target nucleases to very specific sites in the genome and to create those double-stranded breaks. And we're going to start here on the left with, I'd say, probably the most popular and arguably the most famous of these engineered nuclease systems, and that's the CRISPR-Cas9 system. CRISPR stands for clustered, regularly interspaced palindromic repeats. Absolutely no need to remember that, but it's an RNA-guided DNA nuclease system that was actually identified as part of a bacterial immune response pathway, more specifically in Streptococcus pyogenes, as you see here on the right, where we know that they're an adaptive part of the immune system that helps protect these organisms against invading DNA. What modern-day scientists did, though, is they basically understood and gained an understanding, an application of this molecular pathway, and then co-opted it for use in eukaryotic engineering. And so what you need for CRISPR-Cas9 systems is an RNA-guided DNA nuclease system, right? So bacterial enzymes that cleave double-stranded DNA with the nuclease or enzymatic activity conducted by an enzyme called Cas9. And the way I was taught of it was, think of Cas9 as a pair of molecular scissors, if you will, right, that can cut across those two strands of DNA at a very specific location in the genome. And what that does is it enables bits of DNA to be added or removed. At the bottom here, we also have a piece of RNA called a guide RNA, which is something that's designed to find and then bind to a specific sequence in the DNA. The guide RNA, as you might imagine, has RNA bases that are complementary to those of the target DNA sequence. What that means, at least in theory anyway, is that the guide RNA will only bind to the target sequence and no other regions of the genome. And so this guide RNA, as you see on the left here, or gRNA, consists of a really small piece of a pre-designed RNA sequence, typically about 20 bases long, that's located within a longer RNA scaffold. The scaffold part binds to the DNA and then guides or recruits that Cas9, those molecular scissors, to the right part of the genome based on a pre-designed sequence, making sure, ideally anyway, that the Cas9 enzyme cuts across both strands of DNA, again at exactly the right spot. Now, the Cas9 complex actually steers and integrates a small piece of foreign DNA, something called a PAM, which you might have heard of, or a protospacer adjacent motif, into the CRISPR loci in the chromosome. And for S. pyogenes, in this case, this PAM motif is always an NGG sequence. So the PAM essentially acts as a binding signal for the Cas nuclease, allowing it to distinguish between foreign and host DNA, and therefore ensuring that the CRISPR locus itself is not targeted and destroyed. So really big picture here, the take home is CRISPR-Cas9 is a novel gene editing system. The system consists of two parts, right? The Cas9 enzyme and the guide RNA, which allow us to modify, edit, delete, or even correct specific areas of our DNA. Again, all by quite cleverly capitalizing on an endogenous repair mechanism. What happens next, though, I think is really quite interesting because, as I said, the cell actually recognizes that DNA is damaged, and then it tries to repair it, right? And so we have here a cartoon representation of that, of what's happening at the molecular level. In red at the top here, we have a double-stranded piece of DNA, right? And the gray circle represents an engineered nuclease that's going to come in and cleave that DNA, thereby causing a double-stranded break. When that break occurs, the cell is going to attempt to repair it. And when it does that, there's generally two main pathways, again, or natural repair mechanisms that are involved in this healing process. The first of which is non-homologous end joining, or NHEJ, shown here on the left. And this is the primary pathway for the repair of DNA double-stranded breaks throughout the cell cycle. And we refer to it as non-homologous because the break ends are actually directly ligated without the need for a homologous template, right? So it's non-homologous. These are generally what's used to make, say, knockout models or perform genomic screens. They also occur quite quickly, and they have a high capacity for repair. And again, a lot of that has to do with that there's no requirement for a repair template or extensive DNA synthesis. The second healing process on the right here is called homology-directed repair, or the HDR pathway. And homologous recombination is a bit more precise because it does require a template for repair. And so basically, by providing the cell with a synthetic template containing the sequence of interest, for example, a disease-specific mutation, the researcher can introduce the sequence into the genome. And this is what's done to perform gene insertions or general knock-in editing. For HDR to occur, though, we do require the presence of a donor molecule, one that contains the new DNA that's going to be added to the genome. And as you can see in this figure on the right, those left and right homology arms of that same sequence essentially flank that break site, and that allows the donor template to be incorporated into the genome right at that exact spot. Generally speaking, there's three main materials required here. The first is a specific and efficient nuclease system. The second is a repair template that contains that desired change. And then the third is a method to deliver them into the cells. And so it's really the collection of these materials together that allow the edit to occur at the molecular level. And then last but not least here, I want to point out that there's really four main types of donor templates used by scientists today. I've listed these in the top right here in order of increasing size. The first is a single-stranded DNA. This is generally used for small insertions like SNP changes or amino acid substitutions, typically on the order of less than 200 nucleotides. The second of which is double-stranded DNA. This can be used for a bit larger insertions, things like fusions or reporter genes, typically on the order of about 200 to 5,000 base pairs. The third is plasmid DNA, which is often used for very, very large insertions, up to about 10 kb. And then lastly, viral vectors, specifically things like AAV or adeno-associated virus, which are generally reserved for really big insertions in cells that are quite difficult to transfect. At Thermo, we really utilize mostly the first two, single-stranded and double-stranded templates. Now, another thing that's nowadays especially when you think about different clinical applications, right, with CRISPR-Cas9 systems being so widely used, is that there are, and rightfully so, emerging concerns about off-target events when using this genome editing approach. And these off-target events are something that occur when unintended edits happen, right? And again, this potentially adds another variable or unknown to a particular experiment. I've highlighted here, and again, we'll share these slides like we always do, a few papers along those lines, and just wanted to emphasize that it's important to monitor and obviously try to minimize those off-target effects, especially again for things like therapeutic applications, because this allows us to, in many ways, reduce the risks associated with undesired edits in your experiment. So how do we actually do that? Well, this slide is a nice overview, I think, of a couple of different strategies that are available to help reduce off-target effects. Starting on the left here, we've got an RNP delivery format. These RNPs, or ribonucleotide protein complexes, are basically a protein guide RNA complex. And the way they work is they provide an initial high level of CRISPR-Cas9, those molecular scissors, followed by rapid decay, right? And this transient approach helps minimize the risk of off-target generation. Second strategy in the middle here is something like a high-scoring guide RNA to help us minimize off-target effects. This is a really cool computer simulation program that we offer at Thermo Fisher in the form of what's called our TrueDesign Genome Editor. And what that does is it enables us to predict things like potential off-target loci by providing a guide RNA algorithm score that allows us to predict things like efficiency, specificity, proximity to the edit site, right? And then lastly, the use of things like high-fidelity Cas9. High-fidelity Cas9 is essentially a version of the Cas9 nuclease that's engineered for high specificity or reduced off-target effects. And again, I don't want to go too far into the weeds on this, but the high-fidelity Cas9 is essentially a variant of the Cas9 nuclease, which again, reminder of those molecular scissors, that improve specificity by incorporating additional point mutations. Those point mutations help destabilize interactions between the Cas9 and the target DNA. And that kind of tailored destabilization, if you will, makes the high fidelity Cas9 likely to bind to non-complementary or off-target sequences, therefore resulting in improved specificity. The little table on the bottom left here, when we're looking at comparing the performance between, say, a wild-type Cas9 and a high-fidelity Cas9, it's really this balance or give and take between on-target editing efficiency and the ability to reduce off-target events. So with a wild-type Cas9, right, we expect to see superior on-target editing efficiency, but the trade-off of substantially more off-target events. In contrast with, say, a high-fidelity Cas9, as I just talked about, we have improved specificity and therefore the ability to reduce off-target events, but sometimes with a little bit of a loss on-target editing efficiency. And then the last thing I want to touch on here just quickly with regards to Cas9 nucleases is that they can take many forms, as you can appreciate here on this table. In the early days of CRISPR, scientists often used Cas9s expressed from a plasmid, but that, mostly for efficiency reasons, quite quickly fell out of favor when these newer technologies came about. And so nowadays we often use protein and mRNA formats of Cas9 that allow for DNA-free transient expression without a lot of the residual effects that you'd say see with a plasmid. In our case, the version of the Cas9 is called TrueCut Cas9, and it's quite popular for knock-in and knock-out experiments. Again, if we go back to this kind of high-level overview slide, this time I want to switch gears just quickly and focus on the other system for
Hey,
today.
Tom.
Yeah, go ahead, Omar, please.
Sorry to interrupt. Can I just add in one quick comment on the last slide?
Don't be sorry to interrupt. Please always do, and go for it.
Now, just because I think this is such an important point about off-target effects, and I want folks to really understand the differences here and sort of why we're using those different formats, and they kind of correlate to the length of expression or the length of time that the Cas9 protein is inside of the cell. So these different formats, right? Whether you're delivering the plasmid to the cell, which then expresses the transgenes within the plasmid and then makes a Cas9 protein, right? Versus just delivering the protein itself. The shelf life, the half-life of either the plasmid or the protein here is what's in question. And so a protein will naturally degrade a little bit faster, won't sustain for a prolonged period of time like a plasmid would in the cell. And the reason why that's important, right, is those Cas9 proteins technically can cleave at any point, right? It's a very stochastic process. We use the guide RNAs to guide them to a very specific place in the genome to make a very specific cut, right? The double-stranded breaks that Tom was talking about. But technically, if they're around DNA, there is a chance that they can cleave the DNA, right? So we want to minimize the time that the DNA in the inside of a cell is exposed to that particular protein, which is why, as you know, Tom has mentioned, an RNP complex or a ribonuclear riboprotein, excuse me, complex is where you deliver the Cas9 as a protein and then the guide RNAs as an mRNA to help guide it. But then the expression of those is quickly removed from the cell, eliminating the chance that these proteins are going to keep cutting your DNA. Sorry, I thought I'd just add a little bit color there, but I hope
Absolutely.
that's a little bit helpful.
No, super helpful. Thanks, Omar. All right, I'm going to go back again to this slide and talk about TALENs, transcription activator-like effector nucleases, as we see on the right here. Again, you don't have to remember what that stands like. TALENs stands for. TALENs is fine. But again, these are based on effector proteins that are secreted by certain types of bacteria. In this case, bacteria that we know can cause major diseases in crops. And these pathogenic proteins, basically what they do is they rewire transcription of plants upon infection, and then they induce expression of certain genes, obviously in a much simpler manner in plants. So how does this work? Well, TALENs, as I said, are based on effector proteins that are secreted by certain types of bacteria, specifically the Xanthomonas bacteria. And tau nucleases or effector proteins consist of repeat sequences of about 30 to 34 amino acids. And these sequences or motifs have variable positions, right, that recognize specific DNA nucleotide sequences. basically allowing for unrestricted targeting of any sequence in the genome. And when that happens, right, what happens is the tau effector DNA binding domain binds to this nuclease here called focal down at the bottom here and allows us to form a dimer that induces breaks in the DNA. And that results then in a frame shift mutation and then subsequent genes that results in their knockout. These talons are also now available in an mRNA-based format, so they're quite easy to get into cells like stem cells. And a big part of why they can be addressed anywhere on the genome, because in this case, in talons, there's no PAM motif constraint like we saw with CRISPR. Just quickly, for comparative reasons, talons are often, I would say, limited to more simple mutations, unlike CRISPR, which can introduce multiple gene mutations concurrently with a single injection. CRISPR transfections also have higher efficiency, whereas talon editing often results in a mosaicism, which of course is when a mutant allele is present in only some of the cells that are transfected. Now, as far as delivery goes, that's obviously an important consideration here. And these on the right here are some of the tools that researchers nowadays have at their disposal. As you see, there's quite a few options to carry out that process of gene transfer, as you see in the cartoon. But a good deal of what we employ at Thermo relies on either lipid-mediated transfection or electroporation. So just quickly, we'll start with lipid-mediated transfection here. This is a technique in which we take advantage of the positive charge of lipid complexes, right, to bridge with the net negative charge of the cell and DNA. That complex then enters the cell via endocytosis, as you see here. And there's obviously in this process, like many others, a couple of important considerations, in this case, that impact lipid transfection, things like cell type, passaging number, health, density, the reagent amount and ratio to delivered molecules. But again, all food for thought when designing and implementing an experimental plan. We also do rely quite heavily at Thermo on the process of electroporation, which I've circled down here at the bottom. This is a physical process that uses, as the name implies, right, an electric pulse to create a temporary pore, if you will, in the cell membrane, one that's quite an efficient strategy for introducing nucleic acids into the cells. And again, whether that's for the creation of a stable cell line or just for transient transfection, right? Basically, host cells and biomolecules to be transfected are placed in a conductive solution, right? One that has a charge. We then apply an electric pulse at a set voltage, right? And that disrupts the phospholipid bilayer and results again in that formation of a temporary pore in the cell membrane through which the DNA is able to pass. The pores remain open though for several hours. So we do have to handle the cells quite delicately, even after they've been plated down. And electroporation typically relies on either a cuvette-based technology or I would say slightly more advanced pipette-based strategy, which I'll show you in just one second. And this is definitely, in our hands again anyway, one of the most successful techniques for difficult to transfect cells. Now, again, I won't go into too much detail here, but I did want to outline a couple of gene delivery methods and just showcase a couple of pros and cons of those different methods. Things like lentiviral delivery, microinjection, electroporation, lipid delivery, etc. So just a nice point of reference for you guys if gene delivery is something that you're interested in taking a deeper dive on. Now, quick shameless plug here. At Thermo Fisher, we do often utilize our Neon Next electroporation system for transfections or applications that require physical transfection. This is a really easy to use and fast and very efficient system. You can optimize electroporation parameters, especially to transfect, say, hard cell types, right? It's backed by hundreds of pre-optimized protocols. I think there's about 350 or so that we can use that span about 150 different cell lines. So it's quite a user-friendly way to electroporate your cells. The other thing, a couple of things I just wanted to mention here, because I think they're important. The website at the bottom here is a really nice online tool to search for and actually order pre-designed guide RNAs. It's also a fun website to play around with if you kind of want to muck around in this space a little bit. What it does is it uses an algorithm to help in the design of these guide RNAs for effective gene knockout. And these synthetic gRNAs are available for mouse and humans as well. We also offer human designs for lentiviral guide RNAs. And then lastly here, another fun online algorithm-based tool is our True Design genome editing software, which I mentioned before. This allows you to design an order, populates all the things you might need, your CRISPR guide RNAs, the associated DNA donor. You can add other reagents to the cart directly from the tool in order to create an experiment or complete an experiment. Again, I think it's really fun to play around with. You can kind of create this intuitive visualization of the genomic sequence to sort of help you design and navigate that process, right? So designing complex knockouts, using different donor sequences that can target all over the genome, calling out off-target considerations in the target population of interest. And then one other thing I wanted to mention here, Omar and I specifically cover stem cells and neurobiology. We also have a dedicated scientist on our team, Kevin, who is absolutely brilliant at genome editing. That's his bread and butter. This is all he does. So if this is something that you're really interested in taking a deep dive on, running through different experiments, running through different parameters, talking through in very high detail, very much detailed things like the True Design genome editing editor, we can certainly put you in touch with Kevin. He's a fantastic resource as well. So put a plug in for him there. He's given people quite a lot of success with some of the work that he's done. So good. But if we tie that all together here, this is a nice overview again of the genome editing workflow, in this case, the CRISPR workflow here. So we're looking at the design and build of the workflow, where we're, of course, trying to obtain superior gRNA and donor designs for your experiments. We then pair those, again, with high-performing enzymes to maximize editing efficiency. Culture and delivery is obviously important. We want to get those editing tools into healthy, viable cells with a high efficiency. And then importantly, at the end, things like detection and validation. We want to confirm editing efficiency. We want to know how many cells are modified. We want to validate the edit itself, right? So again, the workflow here is very all-encompassing for design, delivery, and detection, basically allowing you to engineer your cells all from one place. Data-wise, I just want to touch on a couple of posters here that I thought were of interest. This is a recent ISSCR poster, one figure from that, where we demonstrated the high specificity of TrueCut Hi-Fi Cas9 in human iPSCs. And so what they did in this experiment was select a gRNA targeting 3 different genes, HBB1 and 2, which are hemoglobin beta subunit genes that we know are associated with sickle cell anemia, as well as HEC4, which is a well-studied site for off-target effects. So in this TEG-seq data plot here on the bottom left, the TrueCut Hi-Fi Cas9 allows us to register fewer gray dots or off-target events compared to, say, the wild-type TrueCut Cas9 and another competitor, Hi-Fi Cas9 here. And basically, the reduction in off-target events that we observed at HEC4, in this case, represented about 55% elimination of off-target events when compared to the wild-type Cas9. So basically demonstrating that the TrueCut Hi-Fi Cas9, as you see here, enabled higher specificity, in this case, in human iPSCs. Taken from another poster, this is from 2021 ISSCR poster. In this case, we're looking at off-target effects in primary T cells, again with the TrueCut Hi-Fi Cas9 protein. So what they did in this case was selected 21 guide RNAs targeting 4 different therapeutic genes, TRAC-A, CD52, TRAC-B, and PD1, and essentially performed a benchmarking study against the wild-type TrueCut Cas9 and a competitor high-fidelity Cas9, again using TEG-seq as our analysis tool. So in these dot plot graphs here, the red dots represent on-target events, and each gray dot represents an observed off-target event. The dot plot, it's a little complicated here, is based on a ratio of their off-target RPM, or read per million, over their on-target RPM. So the higher the percent ratio, the more likely the off-target events are to occur. And you can kind of appreciate in the graph here that compared to the 2 other enzymes, right, there was a significant reduction in off-target events with the TrueCut Hi-Fi Cas9 across all the target loci in these primary T cells, where we saw the ratio of observed off-target events, those gray dots, right, was relatively lower than other nucleases, again, indicating that these off-target events had a lower probability of occurrence. Now, one other thing, and Omar talked a little bit about this last time around, but another important workflow consideration when we're especially talking about things like gene editing is media choice. And I'm totally admitting here, there's a lot of media choices, right? even within the same company. Sometimes it's totally overwhelming. But I think it's nice in nowadays anyway, in modern applications, to be able to choose specific media that work best for specific applications. So again, I put this in as a nice point of reference here in the context of genome editing. As you see at the bottom here, we universally recommend StemFlex in our case, which offers a lot of support and recovery for cells during stressful applications like genome editing. And again, that's because of StemFlex's strength and versatility during different challenging applications like genome editing and some of the benefits like you see here on the slide. Data-wise, Omar talked about this a little bit, but this is data to support the claim that StemFlex performs well in the context of genome editing. What we're looking at here is recovery after singularization and electroporation with Cas9 protein in the guide RNA. In these experiments here, the cells were seeded at the same density throughout in 24-well plates and then allowed to recover in different media, plus or minus a Revitacell supplement. Revitacell is something that helps with recovery and overall cell viability. So this is an application where we would expect cells to require the addition of something like Revitacell or another ROCK inhibitor to allow for survival of those so-called stressed out cells. But what we found here was that StemFlex showed optimal recovery out of the 3 media that we tested here, even in the absence of Revitacell. Again, this is a bit of data here that I think Omar may have showed before, but it's comparing recovery in different media systems, basically just showing us that, again, media is an important consideration for these challenging workflows. And at the end of the day, you can save a lot of time and money and frustration by picking the right culture system for a particular application. Again, just to put some of that context of gene editing into action, I want to now shift a little bit into demonstrating the utility of one of these systems. And so here we're making stem cells from either healthy donors or patient donors, and then evaluating disease-specific SNP changes, those single nucleotide polymorphisms, again, which are DNA sequence variations that occur when nucleotides, right, so adenine, thymine, cytosine, guanine, are altered in the genomic sequence. We either corrected for those SNP changes or introduced a mutation into healthy donor cells to reproduce disease phenotypes in this case. And so Cas9 iPSCs were grown in StemFlex medium. We then used our GeneArt kit to support CRISPR editing, right, where we made guide RNAs to make SNP conversions at different locations or genes. We then delivered those guides alongside a single-stranded DNA oligo donor using the system I talked about, the neon electroporation system. We then measured cleavage using the genomic cleavage detection kit. And then on the top here, right, is a gel representing results from genomic cleavage, basically showing that double-stranded, double bands appear as you increase the amount of guide RNA. That indicates a product that results from genomic cleavage, which we would then quantify. In the bar graph here, you see percentages of insertion or deletion, right, also known as indels in blue, or HDR, homology-driven recombination in red, for a bunch of different loci associated with different diseases. And these include things like central nervous system disorders, epilepsy, schizophrenia, Parkinson's disease are all on here, as well as mutations associated with cardiac rhythm disorder and dilated cardiomyopathy, which I think is as good a segue as any for our case study here. So last but not least, I'm going to expand a little bit here and talk about dilated cardiomyopathy, which is essentially when the heart chambers enlarge, right? And they enlarge in that process of enlargement, basically lose their ability to contract. So this is one of the many disease modeling success stories that we have through collaborations between our customers and our R&D team. As I said, it also, I think, sets the stage quite nicely for next week when we're going to be talking about mesodermal differentiation. And this is all in 2D, but Omar is going to be talking to you about some really cool space that we've done in the 3D space as well. He's got some really pretty pictures to show you of beating cardioids, one of which picture in particular, and Omar knows the one I'm talking about. It's absolutely mind-boggling. But what we have here is a specific heart disease model where you can see kind of in this overview here, we're going to go through, again, the different buckets of the stem cell workflow. Using a Cas9 expressing iPSC parental line, what they did here is engineered a mutation in something called the troponin T2 gene. specifically a TNNT2 mutation. A TNNT2 mutation often leads to dysregulated muscle contraction in the heart, and that's due to alterations in intracellular calcium. And this calcium dysregulation is an essential component of that disorder of dilated cardiomyopathy. We know that, as I alluded to in humans, dilated cardiomyopathy usually starts in the heart's main pumping chamber, the left ventricle, which, as I said, over time stretches. And when it stretches, the tissue thins, right? So it dilates when it's trying to do its job, when it fills with blood. And it therefore loses some of its strength and integrity, and it can't pump the blood as well as, say, a healthy heart. So what we did here is, after generating this model and subsequent genome editing, we characterized the engineered iPSCs. We differentiated them into cardiomyocytes using our cardiomyocyte differentiation kit, which, again, Omar will introduce a little bit more next time. And then we performed a bunch of different cell-based assays to understand the effect of this mutation on the cardiomyocytes themselves and heart cell function. From that cardiovascular disease modeling workflow, looking just a little bit more closely here at the characterization process, after introducing the mutation into the Cas9 iPSCs, what our team did is reconfirm targeting of that troponin T2 gene by using Sanger and next-gen sequencing. And again, without going into too much detail here, they examined the quality of those edited iPSCs using a couple of different characterization tools. Importantly, again, this allowed them to confirm that things like pluripotency and karyotype in the cell line were essentially unaffected by genome editing and line clonality in this case. After they confirmed the genomic edits, the Cas9 parental controls along the bottom here and the mutant lines were differentiated into cardiomyocytes, as I said. I think you can appreciate here that, similar to the Cas9 parental line, the troponin T2 mutants differentiated normally. We can see this is evident here with both the mutant and the control lines down here and the mutant at the top, standing appropriately for troponin 2 in red and actin 1 in green, both of which are structural proteins and essential components of what will be the contractile apparatus in cardiomyocyte cells. So this basically indicated successful differentiation of cardiomyocytes from the troponin T2 mutant line and the Cas9 control iPSCs. What they did next is examine functionality, which is obviously important in the setting of an organ like the heart, in the mutant cardiomyocytes by monitoring calcium transients, right? And so, again, under normal circumstances, calcium transients in the cytosol play a really important role. They control each cycle of contraction and relaxation in the beating cardiomyocyte. And as you could probably imagine, right, with the mutation, what they saw here is they observed quite a readily apparent delay in calcium decay over time in these mutant versus control cardiomyocytes. You can see this in the top left graph here, where they used a dye, something called fluo4, to visualize cellular calcium levels in both the mutant, which we see in orange, and the control, kind of bluish purple cardiomyocytes. This delay in calcium transients also correlated with a slightly lower beat rate in the mutant cardiomyocytes as compared to the wild type controls, which is demonstrated up here in this top right panel. And then lastly, under stressful conditions, this is, I think, where this experiment does really well because it's applicable, right? We think about things like extensive exercise, for example. That's quite a stressful condition for our heart. And what happens in those settings is heart cells or cardiomyocytes increase the beat rate, right, to help support increased blood flow. And so along those lines, cardiomyocytes in culture can actually be stimulated with a beta-adrenergic drug, in this case called isoproterenol, to accelerate their beat rate with something called DMSO, which does not induce this effect, this accelerated beat rate, which we use as a control. And so we use this system, right, to mimic cardiac stress in vitro. You can see this down here in the panels on the bottom left, where, not surprisingly, spontaneous contraction in the troponin T2 mutants was quite rapidly compromised after isoproterenol administration. So just to summarize quickly, this case study here, again, we used a Cas9 expressing iPSC line to generate this troponin T2 mutation. We then confirmed that the edited iPSCs maintain things like pluripotency and a normal karyotype. And then we differentiated those edited iPSCs into cardiomyocytes. And in this case, we discovered that the mutation affected spontaneous contraction rates, often leading to challenge-induced cardiac arrest, and therefore mimicking some of the pathophysiological hallmarks of the disorder. We're talking about dilated cardiomyopathy. So I'm going to wrap this up here. But again, I think we're in such a good space and such an interesting space as well, working in stem cells. We've got all these new technologies at our fingertips, which is so, so cool, all the way through the workflow, right? For the generation of iPSC systematic cells that allow us to nowadays create these limitless pools, if you will, of iPSCs. The iPSCs obviously contain some of the genetic makeup of the cells from which they were derived, but we can then use them with tools like gene editing, right? CRISPR-Cas9, for instance, to generate things like knockouts, to study different genes in cellular processes. We can look at knock-ins to think about things like point mutations in different disease states, reporter cell lines, right, which obviously have a bunch of different applications as well. We talked a little bit today about electroporation and lipid-based technology methods, which allow us to generate edited iPSC cultures. Again, obviously in this case under feeder-free conditions in 2D, which in our case we found to be optimal with StemFlex medium. And then we showed that these iPSC and CRISPR-Cas9 systems were quite a good tool for influencing different mutations, and in that the functionality then of cells, right? In this case, especially cardiac cells in the context of dilated cardiomyopathy. Again, I put this slide here just as a quick overview as to what we talked about today. It's a nice point of reference, I hope. There's also, again, some really cool resources down here at the bottom. We actually have a really, really good genome editing website that has all the different tools that I talked about today. If you're interested in playing around or looking some of these or just keeping them in your back pocket moving forward. Again, we also have a dedicated scientist who's really, really, really sharp on all this stuff on our team as well. So we can certainly provide his contact information if that's handy. But again, I'll stop there in the interest of time. We really appreciate you guys joining us for all of this. I hope the course is going well. Please give us any feedback, good or bad. We'd always be happy to take it. This is something that Omar and I really, really enjoy doing. It's a good opportunity for us to get out and talk to loads of different people doing all kinds of different things that we are not necessarily privy to at Thermo. You guys are the future of what's happening in the stem cell space. So thanks for letting us be just a small part of your journey. But thanks for joining, and we'd be happy to take any questions.
Thank you, Tom. That was great. Yeah, so if anybody has any questions, please drop them. Come off mute and ask them if you want to. And while folks are doing just to kind of echo what Tom was saying too, it is really, really big space at this point. You know, we talked about over the last couple of lectures about how far we've come as a field, right? And this is just an extension of the cool new different tools that we were now able to use with these particular cells, right? Making genome edit in pluripotent stem cells specifically here. And I say this because if you guys look back maybe 10 years back or so, it would take I don't want to date myself, but this would be an entire like doctorate thesis, right? Of making one edit in one gene, particular gene, or creating one transgenic mouse line, for example. Used to be a 5-year project, and now all the progress that we've made, you saw all the tools that Tom was talking about. This could be a process where you can generate a cell line potentially within 6, right? That being said, you really, really have to consider a lot of the small things. And Tom kind of mentioned really, really helpful hints. I hope you guys take with you as you move forward in planning those gene editing experiments. They are nuanced. It is, the smallest details may potentially be something you want to watch out for, right? Whether you're using an RNP complex, like protein itself, or plasmid for delivery. Is it electroporation or lipid transfection? Which media are you using? Which matrix are you plating back on? There is a lot of work that's been done already that you don't need to repeat. So please feel free to ask for protocols. Go talk to your cores that have a lot of experience with some of this stuff already, right? And we're here to help, like I mentioned. And as soon as I get off my soapbox, which you know I always like that, we have a couple questions. What is the cost of your R&P system? How many experiments do you get per true cut? Honestly, Angel, I have no idea. Tom and
Great.
I, yeah, Tom and I don't really like know anything about the sales side. But we are happy to put you in touch with somebody that can get you that information specifically. So I can, Tom, we can figure this out between the two of us.
Yeah.
We can send Angel some information there. And then Emily also you mentioned handling cells after electroporation. What things are you consider practicing? That's a great question, Emily. So, you saw that there's a couple of considerations there, right? So, one is the delivery mode itself. Tom talked about lipid transfection and electroporation as the two main ways we use, right? Electroporation is usually more efficient at delivery, but it's a little bit harsher because you're creating those pores in the cell membrane. So from a recovery standpoint, getting these cells back into your culture dish as quickly as possible, and allowing them to go back into 37 degrees in a media that would be very supportive, right? So as opposed to something like Essential 8 that would be very lean, having more support, you know, like StemFlex, for example, which is what Tom mentioned, would help these cells quite heavily. We also add ROCK inhibitors at that point to get the cells to rearrange their cytoskeleton and increase surface area a little bit more, which allows them to attach a little bit better, recover a little bit faster. So you can use the Bravita supplement. Recently, there is a new supplement from that came out from the NIH called Sept or Culture Sept, which is something that I would recommend, a ROCK inhibitor, but also some pro-survival anti-apoptotic small molecules. Um, what else? And with the neon specifically, right? Like even the electroporation instrument that you're using, for example, the neon the electroporation instrument we have is a neon. The zapping of the cells or that electric pulse happens inside of a tip that's very thin, where it was designed that way on purpose as opposed to a cuvette, so that you can have a very uniform and simple electric field that these cells are exposed to. And so once that happens, we can, from that tip, just dispense them directly into the culture plate. Again, this is an attribute of the actual instrument itself, but it can cells recover, right? That's why we're really like using the neon because the cells really recover pretty well afterwards. But yeah, things like that, you know, are to keep in mind. But you know, there's other smaller things like that we didn't mention, but I'll give you one example. The different buffers, for example, Tom. Um, the electroporation process, right? Depending on the cell type that you're zapping or that you're delivering stuff into. In our case, it's pluripotent stem cells, but you could use this instrument for pretty much any cell type. Um, but when you do that experiment, the strength of the pulse, right? How much energy you're supplying and how long you're zapping the cells for and how many times is something that you can actually control. Whereas, you know, you want to make bigger holes or smaller holes in that cell membrane. And when you're actually doing that, right, you have to really consider what these cells are sitting in at the time, right? Tom mentioned that where we have them in an electric or electrically conductive buffer at the time, right? So with the neon, we offer three different buffers that come with that kit, and you have to figure out which buffer best suits your cell type at that particular you know, voltage that you want to put them in. There's one buffer that's good, like general purpose buffer, but there's also one that would be really good for knock-in specifically experiments. So, you know, these little things can make a big difference in how your cells recover.
Yeah, also just a brief introduction of the available resource in UCLA, Human Stem Cell and the Genome Engineering Center. We received a sound grant of about $4 million, and we collaborate with the Thermo Fisher automation team. and then they are going to establish the robotic high throughput automation for the iPS cell generation and then gene editing, and then also tissue differentiation. So next week, we are going to talk about mesodermal differentiation like muscle, and then also special guest, Dr. April Pyle will present, and the following week is neural differentiation, also a special guest talk with Dr. Michael Well. And then also we can do something like 100 different clone of the something like this. Just to think about a new design of the experiment. Without robot and the automation cannot do. Can we make something like 100 different clone of the tissue differentiation with your hand? Or
It's gonna
100
take
different,
so
yeah,
much time.
yeah,
I would not want to do
gene
that.
editing for more than 3-digit level of the 200, 300 gene editing, or 200, 300 iPS cell generation. And with the human hand, yes, we can do it, but very difficult and boring. So you would better use your brain power hand with a high quality job, and then lower quality repeated job, let robots and the automation do it. That is another resource, and then also another way to develop your new idea develop. Just to think about the 100 3-digit level of the independent different clone, and then certain lineage of the tissue differentiation, muscle, brain, T cell, whatever you want. And then robots can adapt what you want. Also, 100, 200, 300 iPS cell generation, as well as gene editing. Yeah.
And Keita, I can't endorse this enough in terms of reducing the labor intensive work.
Yeah.
These kinds of things we typically, after you do your gene editing experiment, right?
Yeah.
One thing that maybe we didn't talk about as much, but we kind of mentioned before, once you do the gene editing experiment, right? You zap your cells, for example, deliver your CRISPR-Cas9, your guides. The process of the editing is very stochastic, right? It's by chance, and it can happen on one allele or both alleles or none of the alleles, right? So after your that gene editing experiment, you're going to have a pool of cells, some of which will have your edit, some of which will be homozygous, some of which will be heterozygous, some of which will not have the edit at all, right? They'll be wild type. And you have to, for a lack of a better word, sparse them out and clonally expand individual ones to see which edit that you want, right? And this is where the use of automation robotics makes it so much easier to pick which clone is the one you want to be working with, right? If you want to make a gene edit, you don't want to be working with a wild type cell line or even of both wild type and mutant cell type cells together. You just want the mutant cells, right? And that takes a little bit more effort to dig those clones out specifically. So good call out for sure. If you can use or make your life easier, why not?
Yeah, also just new idea development, something like with this equipment or system, and then before we cannot do certain type of experiment. Now with this, and then we can do it. And then also it will reduce the cost because labor is one of the biggest part of the your PI or PI should consider. And then within 1 hour, can we do what type of the experiment? Yeah, very limited. So therefore, cost reduction is also one of the key part. And also, what is the most important part is that everyone attending here is really, really valuable, highly educated personnel. And then I feel sorry just doing the repeated job. And the more work on the more hypothesis develop, more true science is always better. Yep. Okay, anything else we need to discuss?
Think we're all good. Any more questions? There was one more question about which buffer is the knock-in buffer. It's a buffer called the genome editing buffer that's included already in all of the Neon kits that you would purchase. It's one of three buffers actually that comes with the kit. There's the R buffer, T buffer, and the genome editing buffer. Um, but again, like I said, the difference here is between the genome editing one as opposed to the other two is that the genome editing buffer blocks non-homologous end joining, and for a short period of time that allows for a period where you can do homologous recombination, right? A little bit easier. So you're able to incorporate the template that Tom was talking about. For the R and T buffers, those are a little bit different. They're based on the conductance of electricity for the two different buffers. So depending on the cell type they're using, they may prefer one buffer over the other, depending on the voltage that you're going to be using or zapping these cells with.
Yeah, next we are going to talk about mesodermal differentiation. And then just to think about what is the mesodermal lineage. Because, yeah, I'm not interested in mesoderm or muscle differentiation or whatever. No, that is not true. Actually, the one tissue type differentiation is actually track down all of the evolution, and then also what is that, the lineage differentiation. So therefore, mesoderm, including what? Blood or anything. Initial stage of the tissue differentiation is identical. And then after that, they just branching out, use different cytokine, different method. So therefore, even if you are not in the mesodermal tissue differentiation, always how they develop the protocol and how they adapt this is really, really useful to change your type of the cell type when you differentiate. So therefore, just a broader knowledge like endoderm, mesoderm, ectoderm always is useful. So therefore, please do not underestimate the contents of the next lecture. And then also there is a special guest, Dr. April will give this research and then also perspective. It will be really, really useful information for you.
Yeah,
Okay,
I
good.
agree. Perfect. Anything else, Tom?
No, I think that's it. Thank you
Okay.
guys. Appreciate you joining.
Hey,
Thanks,
bye.
everybody.